Thank you. What Anthony didn't say was my interest in being here, and the reason I offered to speak was because I have been part of a clinical trial, and I have both a scientific background, a business background, and of course I'm a consumer. My hope today, my plan today, was to actually share with you the good news about a novel new treatment. And what I'm going to be doing is I'm going to tell you about my experience, the discovery and the research that I went through to learn about ocular melanoma, the treatment, and then the crossroad that I'm at right now. Today will sort of go down in my mind as sort of my coming out. Ocular melanoma has not really been part of who I am. I have a lot more that I want to do in my life. It's not really part of my identity, and I haven't really been telling anyone about it. So this is the first time that I've actually spoken. It's the first time that I've given this presentation. The food was so good yesterday at lunch that maybe I should call this my coming out party. And I've annoyed Alex a couple of times. By the way, thank you, Alex, for organising such a great event. I've sent him my presentation 3 or 4 times because I couldn't quite settle the opening slide, which is right now, and this is how it was yesterday. My plan today is to share with you the good news, but I'm not sure, in fact, whether it is my plan today was to share with you the good news, and I'll tell you a little bit more about why I'm not sure whether it's an is or a was. So, my first contact 7 months ago went like this: you have eye cancer, worry first about your life and then your sight. And my reaction was, what? I have no symptoms. And then the next thing was, well, there's really no cure. And the treatment that we have for this is 50 years old. And this is the little devil. This is my new friend. This is the thing that I have because I'm Australian, I have blue eyes, I have freckles, I'm a white male. It's really small. It's sort of the thickness of the end of a crayon and the width of a little rubber on the end of a pencil. But it's now part of who I am. And to try and feel better, I did what most of us do these days. I went to Amazon to buy myself a present. And this was the jigsaw puzzle model that I ordered. I have no idea why they called it 4D. I think maybe 4D for dummies. It's 32 parts. Down the bottom it told me that it was going to be a 30-minute thing to put together. And I very proudly put it together in 2 hours. I did feel like calling you up, Max, during the whole sort of annoying process of putting it together. And I did bring it for you guys today to show you that I really did put it together. And as one of my kids said to me, Dad, is that thing on the top the tumour? And I didn't call you Max because I know that I took up so much time in your office asking you lots of questions. I thought it would be completely out of line if I called your reception and said, I need Max's help to put together this eye puzzle. Anyway, for those of you who don't recognise that brown thing on the top, that's, I don't know the technical name, but I know that it makes tears. So anyway, I put it together and it made me feel like I was making progress. I'm running a business in the US, so I'm very comfortable in the US. I was originally born in New York. I said to Maxine, who's here, my lovely wife is here, say hi. And I stopped my life and went on a little bit of a tour of US cities to meet with the experts around the world on ocular melanoma. And we're very lucky to have Dr. Max Conway here, who's going to be speaking next, and Dr. Anthony Joshua. They told me about a clinical trial. So sort of on my checklist of things to do, I wanted to find out about the clinical trial. And it was a clinical trial on choroidal melanoma, also known as ocular melanoma, also known as uveal melanoma, and sort of doctors are not very good with Google because with Google it's better to call it one thing, but I did manage to do lots of research and And to make myself feel better, I actually registered all the domains I could find on ocular melanoma. So if anyone would like to buy a domain or would like to be on my website, I have ocular melanoma.co, ocular melanoma.guru, ocular melanoma.news, and maybe I can help Anthony with your site promotion. So these are some of the people that I spoke to and helped me understand about my disease. The one on the left is probably the most important. He's been treating me now for over 40 years. And maybe you don't have your glasses on, Les, but can you stand up for a moment and just say hello? This is Dr. Les Brenner, who has been practicing for over 60 years as a GP and was just recognised with an OAM for his service to the community. And Les was the person who put me in contact with the ophthalmologist who I met with before I met with Max, and then I met with Anthony. And by the way, I should really say big thank you to Anthony, because we are such a tiny little population in a tiny little population, and it is so wonderful to have someone like Anthony and Max who really believe in us and this cause and why it is important. And if it wasn't for Anthony's interest, we wouldn't be here today, and we wouldn't have the focus that we need on this really important disease. And then I met with Michael Gibbler. Michael, are you here today? Yes. So Michael is another one of the extraordinary practitioners and clinicians that we have in Australia. And I love Michael for a particular reason. You'll see down the bottom of all my presentations, it says that I'm Wing Dude, and that's who I am on Twitter. I won't say follow me on Twitter, but I will say that I love flying, and Michael and I share an interest in flying. So when I went to see him to talk about my ocular melanoma, we actually spent most of the time talking about flying aeroplanes. The rest of the people on my list are all people that I met in the US, and quite by luck, one of those people is actually here with us, Leanne. Will you say hello to everyone? Leanne has been practicing with Carol Shields, who is the other guru. Yesterday, Bertel was introduced as the god of ocular melanoma, which he sort of cringes when I say. But if he's the god, Carol's the guru, and Leanne has just been in the US for how long? 1 year with Carol, and is now working with Michael and Max in their practice. So we're very lucky to have that knowledge now in Australia. Down the bottom is Dr. Hans Gross-Nicholas, who led the rabbit study. The rabbit study was all those lucky rabbits who were experimented on before the clinical trial, and I'm going to tell you a little bit about that. And then, of course, you know, the other heroes are, I guess, the patients and the support people. And here I thank my wife again for being so wonderful and supportive. And I, on behalf of all of us too, I thank the doctors who are interested and these supporters who have helped us get through and understand what it is that we are dealing with. So just a moment of revision before I talk about the clinical trial. There are really three classes of cancer treatment, and all of them sort of work in a non-specific way, and they have lots of side effects. And even the new treatment, the immuno treatment, which has really become popular and in some cases successful, work in fairly non-specific ways. And what attracted me to the clinical trial that I wanted to find out about was that it was a very specific treatment. The other thing that attracted me to the clinical trial was the 50-year-old treatment that I did not want to have, that I know quite a few of you have had. And the reason I say I'm at a crossroad is because I have to decide right now whether I continue the clinical trial or whether I actually revert to the more traditional treatment. And the traditional treatment, for those of you, does everyone know about brachy? Yes? For those of you, anyone who doesn't know about brachy? So for those of you, about 10 of you, brachy treatment is a radiation treatment where a metal plate is sewn into the back of your eye with radioactive material on it, and depending on how that radioactive material is placed and the strength of it, it then essentially sort of burns the tumour in the back of your eye and kills it. The problem, of course, is that it does destroy other things in the process. And that's the little operation that only takes 45 minutes, where they put the plaque in your eye and they leave it there for 5 days and then they take it out. This is Dr. Paul Finger. He also is a guru in this space. He's done thousands of these operations. And I was very lucky. He spent a few hours with me. And that nice lady in the blue is Nancy Holub, who was one of his patients 10 years ago. And Paul was sort of being very nice and letting me talk to her about her experience over the last 10 years with brachytherapy and then receiving anti-VEGF injections regularly. Paul showed me this chart. This is probably the most important data that I learned and I shared with my family. This chart tells me what my risk is of surviving, my chances of surviving. Sort of if you land in the middle of that, sort of around a 2 or a 3, it's about 50%, because the thickness and the basal diameter of the tumour determines what the risk level is. Quite fortunately for me, I'm down sort of in the forward right corner of 1. I'm sort of between a 1 and a 2 right now, which means that I currently have an 18% chance of fatality over the next 5 years. And this data is really good data. It was produced by reviewing about 3,000 patients and what happened to them based on the size of their tumour. And I said to my son, Sam, good news, I only have an 18% chance of something bad happening. He said, Dad, that is not a good number. Paul showed me this picture. This showed me where my tumour was. My tumour is actually just in front of the inferior oblique muscle, which means that we need to cut the inferior oblique muscle to actually work on the tumour. It's not a great position. It's near the foveal centre, and it means that when we try to sort of sew the muscle back on, that I may have, I have a sort of greater chance of having some sort of double vision. So all of these things sort of brought me to, well, would I like to be a guinea pig on a clinical trial? And as a scientist, I was quite fascinated with the technology. And so I went on this sort of journey talking to all of the practitioners in the US who were involved in the clinical trial. And the Aura story is a really interesting one. It begins with John Schiller. Those of you who've heard about HPV or human papillomavirus, it was discovered that the human papillomavirus was a sort of precursor in cervical cancer. And that work was done by John. It was also found in a really interesting study on rats about 10 years ago, where they took about 80 different types of tumours, and they found that the HPV virus was attracted to the heparin sulfate group that was expressed on about 80% of these cancer types. And someone came up with the idea of, well, if deactivated HPV virus is attracted to tumour, the surfaces of tumour cells, then why don't we attach to the HPV virus something that will kill the tumour? And so they did that. They attached a very big molecule, and that big molecule was a dye that, when it gets light activated, actually then kills the tumour. The big issue, though, in the US is if we've got a really good idea, how do we actually trial it? And we're very lucky, in a sense, that we're such a small group, because the FDA in the US has said that we can be part of the orphan drug program, which means that people like me can be part of a very small group of about 30 people. and we don't have to do double-blind trials where I end up not knowing whether I'm getting a placebo or not. So this is how it works. It's an injection into the eye. That big blue and pinky thing on the right is the dye HPV conjugate. It attaches to the HS group on the tumour, and then when they shine light on it, the dye oxidizes and kills the tumour. And that's the theory. And I found that intellectually really interesting and appealing, and I then decided that I wanted to try and get on the trial. I met this woman who is amazing. I've mentioned her already. She has, how many children? 6 children, Leanne? 7 children. This is Carol Shields, and she and her husband Jerry have written 13 textbooks on eye disease. They're not all her children on the wall. Those children include Michael. They're all the people that have trained. And Leanne, I imagine you'll have your picture up there one day if you're not there. You're already on there, down the bottom. Excellent. These are all the people that Carol has trained around the world. So Carol and I had a long chat, and to be eligible for the trial, the tumour had to be a certain size. It had to be proven that it was growing. I had to be healthy. Les organised for me to do all sorts of health checks, and after some back and forth, I then qualified for the trial. Yay. And so they gave me the informed consent form. I signed it, and I became part of a group of 6 other people who were on the maximum dose component of the trial. I'm down the bottom in that September 2018 group. These are the people at the Wills Eye Hospital who treated me and worked with me. Sandor on the left, who looks like a Hell's Angel bikie. He's the bloke who took photos, and he's an incredible human being. We became mates. We talked about all sorts of things, including Brexit, Bertel, and the mess. He spent about an hour with me on every one of my visits, and I was there 14 times over 6 months. And he helped me understand exactly what was going on with my pictures, my photos, the images, and the things that we were taking. Then Prasad is the doctor who worked with Leanne in actually recording what was going on, testing me. Aman in the suit on my right there, he administered the injections and did the PDT. And there's Carol, and then it's Juliana, Marie, Rachel on the far right, who runs the program, Lisa, Roma. Roma has amazing hands. Roma would be the one who would hold her hand up while I was getting the injection so my eye didn't move. And then Leslie on the right. And as you can hear, I got to know them all quite well. So I was patient number 900 space 31, and that's the drug there, out of the liquid nitrogen. I had 6 ocular injections and 6 PDT over 3 visits, and then another 10 or 11 follow-ups. This was me in room number 12, waiting happily for my injection. And this is a really big facility. It treats almost a third of everyone in the US who has ocular melanoma. And this room 12 was one of 16 rooms. And when I arrived there at 6 a.m., thinking that I would be the only one, there were 100 people waiting to be treated. That's me after one of my treatments. That's my eye after a day or two after a treatment. And then I've included this little picture to remind me to mention to you guys how lucky we are in Australia with our health system, and I think we all know that, but this is the $400 bottle of drops. The treatment, if I wanted brachy treatment in the US, is about a $45,000 treatment, and then the Aura clinical trial, they expect to sell between $50,000 and $100,000 for treatment. So how lucky are we that we live in this country where we can just get the health services that we get here? So I said this was about a crossroad, and I'm right in the middle of this crossroad right now. because if you look at this, you look at where we are today compared to 6 months ago, the bottom right is what the picture looked like on the 4th of September last year, before the treatment. And then the one on the top is what the tumour looks like now. And you have to look at it for a little bit, but can anyone in the audience who's not a doctor tell me what they think they see? It's grown a little bit. It's grown a little bit in one direction, a little bit to the top, and then it's not grown at all towards my foveal centre. So that's not great. Though what's interesting is that the data tells us that if I hadn't had any treatment at all, on average, if untreated, would double in size every 60 to 220 days. So it's not a definitive result. I've been annoying some of the doctors and experts here. I've followed Bertel around twice yesterday, trying to talk about what should I do now? Because this data is not terrible, but it's not good. And Carol and I, on the day, decided that this was a fail. And just to remind us where I've come from, this is an image back from 2004. This is what the freckle looked like in 2004. And I was told by an ophthalmologist that there was a 1 in a million chance that this would turn into cancer. And then this is what it looked like in 2015. It was a little bit denser, but it was still about the same size. So what we're seeing now is a sort of an increasing rate of growth. And so to finish my presentation, I'm going to talk about what's next. Do I do a second cycle of aura? Do I do brachy? And do I do a biopsy? Brachy I've already talked about. The question for me is, who do I do it with? When I was in Carol Shields' office the week before, she did 11 operations. She's done thousands of operations. Paul Finger has done thousands of operations. We have some amazing practitioners in Australia who I really feel care about me and who are local for me, who have not done as many operations. So I have to decide, do I hang out in a lonely hotel room in Philadelphia, or do I come home and be with my family and caregivers who I know will be around for a long time? I need to decide which isotope is used for the radiation. Some isotopes are much more corrosive or toxic to other parts of my eye, depending on how it's applied, Bertel will say. And then I need to decide about whether I do a biopsy. And the problem with the biopsy is that the biopsy itself can cause a fragmentation, which can cause a metastasis. And if we don't take the right piece of the tumour, because it's a sort of uneven mutation, we might pick a piece of the tumour that has a fairly benign mutation and miss the part of the tumour that could be fatal. So I have to decide whether I want to have a biopsy or not. What's really interesting, and I was delighted, Ellen, are you here still today? So Ellen is from Perth, and Ellen is doing some amazing research on non-invasive techniques to look at circulating tumour cells. And it would be just a wonderful thing if we could do a blood test and tell me what my risk of metastasis is. So I know I'm running out of time. The next stage for me, if I get to that stage, is what do I do about metastasis? So I'm starting to research something called GP100, and Bertel is a consultant for a company called Immunocore, who are doing some very, very interesting work on GP100. If any of you are interested, I can talk about it later in my panel, but I think I'm running out of time. There's sort of the good news. I say sort of the good news. I was hoping to say the amazing news, but thank you to Max, Anthony, Michael, Leanne. The AURA Phase 3 trial will be coming to Australia. So thank you everyone. Doing my day job now, I'm very involved in blockchain, building something called non-fungible tokens, and that's what I'm doing next week, going back to my day job, thinking about what I do next with this. I guess my state of mind is what doesn't kill me makes me stronger, And if any of you are interested in some of the research that I've done, probably the best article I've seen is one that Carol and a couple of people did that was published in Nature that has about 100 references attached to it. And thank you very much. You've been a fantastic audience.